Welcome to the deep dive. Today we are wrestling with a really tough problem in cancer treatment. We're focusing on high-grade serous ovarian cancer, HGSOC. It's known for having a pretty poor prognosis, unfortunately.
It really is. And what's particularly challenging and frankly tragic is how it behaves.
Mm.
Patients often have a great initial response to the standard chemotherapy, the platinum-based drugs. You see tumors shrink significantly.
Right, like there's real hope initially.
Exactly. But then, almost always, the cancer comes back. And it comes back because it's developed resistance to those drugs.
So that first win is really just hiding a deeper battle going on underneath, an evolutionary battle.
That's a perfect way to put it. Treatment failure isn't just bad luck. It's quite literally natural selection happening inside the patient. You have these tiny subsets of cancer cells that either started out resistant or somehow acquired it. They survive the chemo.
While all their neighbors get wiped out.
Precisely. And then they have the space to grow and expand, leading to recurrence. The big hurdle has always been, well, how do you watch this happen? You can't just keep taking tumor samples every few weeks. It's not practical.
Right, you need a window into that evolution in real time, if possible. If you could see which cells are starting to win, maybe you could, I don't know, head them off.
That's the dream. And that's exactly what the researchers we're looking at today tried to achieve. They developed this new method called CloneSeq-SV.
CloneSeq-SV. Okay, so what's the trick? How does it solve that sampling problem?
Well, it's quite clever, really. It combines two powerful techniques. First, they use single-cell whole-genome sequencing, scWGS, on the tumor tissue they get at diagnosis.
Okay, so that gives them the before picture, like a detailed map of all the different cell populations, the clones, right at the start.
Exactly. It establishes the cancer's initial, let's say, family tree. You see all the different branches, the different clones that exist from day one.
But then how do they track changes over time without more biopsies? You mentioned blood samples.
Yes, that's the second part. They use liquid biopsies, simple blood draws. Tumors shed DNA into the bloodstream, this cell-free DNA, or cfDNA.
Okay, cfDNA, we hear a lot about that, but how is this different?
Here's the key innovation. Instead of looking for common mutations, which can be noisy or hard to track accurately at low levels, they designed CloneSeq-SV to hunt for very specific structural variants, SVs, that are unique to each clone identified in that initial map.
Structural variants, SVs. Let's pause on that. Why SVs? Why focus on these SVs, these structural variants, instead of the more common single-letter typos in the DNA code, the SNVs?
Right. SNVs, single nucleotide variants, are much more numerous. But SVs are fundamentally different beasts. They aren't typos. They're more like massive genomic rearrangements.
Okay.
Think cutting out a whole chunk of one chromosome and pasting it somewhere else entirely, or flipping a section around or deleting a big piece. These events create unique breakpoint sequences where DNA that shouldn't be next to each other suddenly is.
So they're like really dramatic, unmistakable landmarks in the genome.
Exactly. And that makes them incredibly specific markers for a particular clone. If you find that exact breakpoint sequence in the cfDNA, you know it came from that specific lineage of cancer cells.
So finding an SV is less like finding a typo and more like finding, I don't know, page 50 stapled right after page 200. It's obviously wrong and very distinct.
That's a great analogy. And this specificity translates into technical superiority for tracking tiny amounts of tumor DNA in the blood, which is often like finding a needle in a haystack.
How much better are we talking?
The error rates are worlds apart. The study found that the background error rate for detecting these SV breakpoints was incredibly low, something like 1 in 10 million reads, $1 times 10 to the 7.
Wow.
Compare that to SNVs, where even with advanced techniques, the error rate was closer to, say, 6 or 7 in a million. $6.07 times 10.
Okay, hold on. So $1 times 10.7 versus $6.77 times 10.6. That means the SVs are nearly, 70 times more reliable, less prone to false positives.
That's the magnitude, yes. It means you can confidently detect that tumor signal even when it's present at extremely low levels without needing complex error correction models that SNV tracking often requires. You trust the signal more.
And is there anything else that makes SVs good markers here?
Yes, there's another key advantage. Often, the SVs that mark these aggressive resistant clones are part of large-scale amplifications. The cell might have tens, even hundreds of extra copies of that specific rearranged DNA segment.
Yeah, so each resistant cell is essentially shouting its presence much louder in the cfDNA because it's shedding many copies of its unique SV marker.
Precisely. It boosts the signal significantly.
Okay, so let's walk through the workflow they used for the 18 HGSOC patients in this study. They start with with that single-cell sequencing on the initial tumor.
Right, the scWGS at diagnosis. Then they need to make sense of all that complex genomic data from individual cells. They used bioinformatics tools, like one called MediaCC2.
MIDI-CC2, what does that do? Build the family tree?
Essentially, yes. It reconstructs the evolutionary relationships between the cells, grouping them into distinct clones based on their shared mutational history, especially those big SV events. It helps define who's related to whom and which SVs mark specific branches or clades on that tree.
And sometimes these defining SVs are really complex, like chromothripsis.
Exactly. Chromosynthesis is that phenomenon where a chromosome or part of one seems to just shatter into pieces and then get stitched back together randomly.
Sounds like pure chaos. How is that helpful?
Because the specific pattern of shattering and repair is unique every time it happens. It creates a hugely complex, bespoke SC signature. It's like a genomic scar that's absolutely specific to the clone that experienced it, an ideal marker for tracking.
A unique, messy fingerprint.
You got it. And crucially, they validated this whole approach. They showed that the amount of tumor DNA they estimated in the blood using these SVs matched up almost perfectly, a correlation of R at 0.95, with estimates using the standard method of tracking mutations in the TP53 gene, which is mutated in almost all HGSOC.
So the platform works. It's reliable. Now, the big question, what did they actually see happening over time in these patients?
Right. This is where it gets really fascinating and maybe a bit sobering. They track these clones through treatment, watching their levels rise and fall in the cfDNA. And they asked, when drug resistance emerges, where does it come from? Is it new mutations caused by the chemo?
Or something else?
It was overwhelmingly something else. The major finding, the real core takeaway, was that drug resistance almost always came from the selective expansion of clones that were already there at diagnosis.
Wait, really? So the resistant cells weren't created by the treatment, they were already present, just hiding?
In most cases, yes. Maybe in very small numbers, sometimes representing only a tiny fraction of the initial tumor population. But they possessed the right genetic tools, the right SVs, the right amplifications to survive the platinum chemotherapy from the outset.
Wow. Okay, so chemotherapy isn't so much causing resistance as it is revealing it. It's like a filter that removes all the sensitive cells, leaving only the pre-existing resistant ones behind.
That's a perfect description. It acts as a powerful selective pressure, a bottleneck. And these pre-adapted survivors then have the whole field to themselves to regrow.
And this CloneSeq-SV method could actually see this happening before the doctors knew?
Absolutely. This is a key clinical point. In 11 of the patients where they could track this, they detected the rise in cfDNA carrying these resistant SV markers, an average of 243 days before the recurrence was officially diagnosed using standard clinical methods, like imaging scans.
243 days? That's nearly 8 months warning.
It's a huge potential lead time.
Why the big delay, though? If the resistant cells are growing, why don't scans pick it up sooner? Is cfDNA just inherently more sensitive?
It's partly sensitivity, yes. cfDNA reflects cell death and turnover, which can happen before a tumor grows large enough to be visible on a CT scan. Scans need a certain physical mass. But it's also the specificity of tracking these SVs. You're not just measuring total tumor DNA. You're measuring the specific clones that are driving resistance. You see the shift in the population towards resistance much earlier. It's detecting the dynamic change, not just the bulk.
Okay, so these winning clones were pre-existing. What kind of genetic features gave them this built-in advantage? What did they have that the sensitive clones didn't?
They saw some recurring themes. Complex events like chromothripsis were often found in the resistant clones, like in patient 0A3. Another feature was whole genome doubling, WGD.
Where the cell just duplicates its entire chromosome set.
Exactly. In one patient, patient 026, the clone that dominated at recurrence had evidence of WGD. And interestingly, they could trace it back to a very minor population of WGD cells that was already present, barely detectable at diagnosis. It suggests WGD might provide some kind of general robustness or evolvability under stress.
And then there were specific gene amplifications, right? That seemed common.
Very common. High-level amplifications, meaning many extra copies of key cancer-driving genes. We saw genes like CCNE1, RAB25, MYC, and OTCH3 repeatedly amplified in the clones that ultimately won out.
Can you give an example?
Sure. Take patient 107. At recurrence, the dominant clone, called clone D, had ramped up its copy number of CCNE1. It had about 13 copies, compared to only 8 copies in the clone that was dominant at the start. Clone D also had this massive 14.6 megabase amplification, a huge chunk of DNA that included the NOTCH3 gene.
So just having more copies of these critical genes gave them an edge.
It seems so. More CCNE1 can push cells through the cell cycle, resisting drug-induced arrest.
Hmm.
More NTRTCH3 can affect various signaling pathways involved in survival, but it wasn't always the same gene.
Ah, so the cancer can be flexible.
Highly flexible. Look at patient 045. Initially, a clone with CCNE1 amplification was dominant, but when the cancer came back a second time, after more treatment, a totally different clone, clone D, took over. This clone D didn't have the CCNE1 amplification.
So what was its trick?
It had instead acquired a high-level amplification of a different gene, RB25. And RB25 is a protein, a GTPase, that's already been linked to chemotherapy resistance in other studies.
So the cancer had multiple potential routes to resistance, and different clones used different routes at different times.
Exactly. It highlights the tumor's heterogeneity and its ability to adapt. What works for resistance at one point might be superseded by a different mechanism later. It really underscores why you need to track these dynamics over time. You can't just rely on the snapshot at diagnosis.
Okay, let me just recap quickly for everyone listening. We've seen that CloneSeq-SV, using these unique structural variants, or SVs, allows researchers to track specific cancer clones in real time via blood tests. The big finding is that resistance usually comes from pre-existing minor clones expanding under chemo pressure, often driven by features like chromothripsis, whole genome doubling, or key gene amplifications like CCNE1 or RB25. And this tracking gives a significant early warning before clinical recurrence. So the question for you listening is, if resistance is usually already there, hiding in plain sight at diagnosis, what does that mean for how we should approach initial treatment? Does it just clear the easy targets, leaving the tough ones untouched? This ability to track clones isn't just scientifically interesting, it has direct clinical implications. The case of patient 044 really drives this home. Tell us that story, it sounds like a real success.
It's a fantastic example of how this evolutionary tracking can guide therapy. So patient 044, at diagnosis, their tumor had two main clones identified by SCWGS, let's call them clone B and clone E. Clone E was pretty standard, genomically speaking. But clone B was really striking. It had an extremely high-level amplification of the ERBB2 gene, which you might know better as HER2.
Ah, the same target as in some breast cancer.
Exactly, and not just a few extra copies. Clone B had an average of over 30 copies of ERBB2 per cell, highly amplified. Clone E, meanwhile, had the normal number. It was ERBB2 wild type.
So two distinct populations. What happened after standard chemo?
The frontline platinum chemotherapy did its job against the standard clone. It effectively wiped out clone E, the ERBP wild type population, but clone B, the highly ERBB2 amplified one, survived.
The chemo selected for the ERBB2 amplified clone.
Precisely. When the patient's cancer recurred, the CloneSeq-SV analysis of their cfDNA showed that the recurrence was composed entirely of clone B cells. The ERBB2 wild-type clone was gone.
So the resistance mechanism was clearly linked to this massive ERBB2 amplification.
Undeniably. And because they knew this, because the evolutionary tracking identified the specific vulnerability of the surviving cancer, the clinical team made a targeted decision. They treated the patient with a drug called trastuzumab deruxtecan, or TDXD.
Which is an antibody-drug conjugate targeting HER2-ERBB2. Right?
Exactly. It's designed to kill cells that overexpress HER2.
Mm-hmm.
And the result? The patient had a complete and durable response. They remained disease-free almost 3 years later at the time of the study publication.
Wow, that's genuinely using evolutionary insights to choose the right drug at the right time. A personalized adaptive strategy.
It's the paradigm shift this kind of tracking enables, moving from a one-size-fits-all approach to one guided by the specific evolutionary trajectory of that patient's cancer.
And it works for other resistance mechanisms too, like resistance to PRP inhibitors.
Yes, another classic example came from patient 009. This patient had a known inherited mutation in the BRCA1 gene.
Which makes the cancer initially very sensitive to platinum drugs and also to PRP inhibitors, right? Because BRCA1 is involved in DNA repair.
Correct. So the patient responded well to chemo initially, and then went on to maintenance therapy with a PRRP inhibitor, which is standard practice for BRCA-mutated ovarian cancer. But eventually the cancer recurred.
And CloneSeq-SV tracked the clone responsible.
Yes, the recurrence was dominated by a specific clone, clone F. When they analyzed the cfDNA and post-recurrent samples from clone F, they found something very telling: a new mutation in the BRCA1 gene.
A new mutation? What kind?
It was what's called a BRCA1 reversion mutation, specifically a large deletion about 1.37 kilobases within the BRCA1 gene. This deletion cleverly excised the original harmful germline mutation.
And stitch the gene back together in a way that restored its normal reading frame, its normal function.
Exactly. So the cancer cell effectively fixed the BRCA1 defect that was making it vulnerable to the PRP inhibitor. It evolved a way to switch BRCA1 back on.
That's incredible. A known mechanism of PRP inhibitor resistance, caught in the act by tracking the specific clone that acquired it.
And importantly, this reversion mutation wasn't there at diagnosis. It emerged under the selective pressure of the PARP inhibitor therapy. Again, it highlights why longitudinal monitoring is so critical to catch these acquired resistance mechanisms.
So we know which clones are winning thanks to their SVs. But how are they winning? What are they actually doing differently at the cellular level? This is where the RNA sequencing comes in, right? The cRNA-seq.
Right. Knowing the genomic changes is crucial, but linking that to cell function and behavior is the next step. They performed single-cell RNA sequencing on some samples and used a computational method called tree-align to map the gene expression data back onto the evolutionary tree defined by the DNA changes.
So you could see the job description or the phenotype of cells belonging to specific clones.
Pretty much, yes.
Yeah.
And they found significant differences.
Yeah.
Even within the same patient, different clones could have very different transcriptional programs active.
What did they see in those resistant clones we just discussed? Patient 009, the PARP inhibitor resistant one.
In patient 009, the resistant clone F, the one with the BRCA1 reversion, showed signs of an increased epithelial to mesenchymal transition, or EMT program.
EMT. That's often linked to cells becoming more invasive, more stem-like, and often more resistant to stress, including chemo.
Exactly. They saw higher levels of classic EMT markers like vimentin, VM. Interestingly, these clone F cells also showed a lower fraction of actively cycling cells.
Ah, so they weren't dividing rapidly, more like hibernating persister cells.
That's what it suggests. A state often described as drug-tolerant persister cells that slow down their cycle to weather the storm of chemotherapy, ready to regrow later. EMT programs are often linked to this state.
Okay, and what about patient 107, the one with the CCNE1 and NOTCH3 amplifications?
For patient 107's resistant clone D, the RNA data confirmed the genomic finding. They saw significantly higher activity in pathways related to NOTCH3 signaling and also VEGF signaling.
VEGS, vascular endothelial growth factor, involved in blood vessel growth.
Yes. And this makes sense because the high copy number of the NOTCH3 gene likely directly boosted NOTCH pathway activity. The higher VEGF signaling is also interesting because this patient was actually receiving maintenance therapy with bevacizumab.
Which is an anti-VEGF drug designed to block angiogenesis.
Right. So it seems clone D, thanks to its specific genomic amplifications, might have had such intrinsically high pro-angiogenic signaling that it could essentially overcome the bevacizumab treatment, contributing to the relapse. The gene dosage effect directly translated into a functional resistance phenotype.
Okay, so we've seen pre-existing resistance, acquired resistance, and even hints at the functional states of these resistant cells. But one crucial question is, was this really selection? Or could these clonal shifts just be random fluctuations, genetic drift? How did they prove selection was the driving force?
That's a critical point. Observing a change doesn't automatically prove it was driven by differential fitness. So they turned to mathematical modeling. They used a type of model common in population genetics based on the Wright-Fisher process.
Wright-Fisher. That simulates how gene frequencies change in a population over generations, right? Accounting for random chance.
Exactly. But they made it much more sophisticated. They didn't just use a generic model, they tailored it to each patient. Crucially, they incorporated the patient's own CA125 levels over time.
CA125, the ovarian cancer tumor marker? How did they use that?
They used the changes in CA125 concentration during treatment as a proxy for the strength of the population bottleneck, the intensity of the selective pressure from chemotherapy. When CA125 dropped sharply, the model simulated a massive die-off of cancer cells.
Ah, so the model incorporated the real-world dynamics of treatment response for each individual patient.
Precisely. Then they ran simulations under the assumption of neutrality, meaning they simulated what would happen if all the clones had exactly the same fitness, and any changes in their frequency were purely due to random sampling effects during those bottlenecks.
And then they compared those neutral simulations to what they actually observed happening with the clones in the patient's cfDNA.
Yes, and the result was very clear. In 11 out of the 16 patients they could model, the observed rise of at least one specific clone was statistically inconsistent with the neutral drift simulations. Its frequency increased far more dramatically than chance alone could explain.
Meaning it had to have a fitness advantage under treatment. Positive selection was actively favoring it.
Undeniably. The math confirmed that treatment wasn't just randomly killing cells, it was actively selecting for the clones best equipped to survive it.
The fittest survived, and it wasn't just luck.
Correct. And this selective process had a clear impact on the overall ecosystem of the tumor. They measured the clonal diversity using a metric called Shannon entropy.
Like measuring biodiversity in a rainforest, but for cancer clones?
Sort of, yes. A higher entropy means more diversity, more different types of clones coexisting. They found that in 16 out of 17 cases, the Shannon entropy decreased significantly at the time of recurrence compared to diagnosis.
So the treatment acts like a fire, wiping out most of the diversity and leaving only one or a few dominant fire-resistant species behind.
That's exactly the picture. Frontline therapy acts as that strong selective bottleneck, drastically reducing complexity and allowing the expansion of only those clones with the specific genomic features conferring resistance.
This really confirms that HGSOC recurrence isn't just regrowth. It's an evolutionary process driven by differential clonal fitness under the pressure of our therapies.
It moves the understanding from theory to observed reality in patients. We can see clones with, initially, maybe only a slight edge gain a massive fitness advantage when the environment changes due to treatment.
So what does this all mean? Where do we go from here with this kind of knowledge?
Well, I think we've clearly shown, building on this research, that tracking this clonal evolution is actually feasible. Using SVs and cfDNA gives us that real-time window. We can watch resistance mechanisms, whether it's a massive ERBB2 amplification present from the start, or a BRCA1 reversion mutation acquired under PRP inhibitor pressure. You can see them emerge and expand.
It feels like it opens the door to a completely different way of thinking about clinical trials and treatment. Instead of waiting for the cancer to show up on a scan, for clinical resistance to be undeniable.
Could we act sooner?
Yeah, could we intervene based on these early cfDNA signals? If CloneSeq-SV shows a clone with, say, that ERBB2 amplification starting to creep up during chemo, could you pivot to TDX before a full-blown recurrence?
That's the ultimate goal of adaptive therapy, isn't it? Using the evolutionary trajectory to guide treatment changes proactively, not reactively.
Or maybe even cycle therapies. If you see clone A rising under drug 1, switch to drug 2, which targets clone A's vulnerability, then maybe switch back if clone B starts to emerge.
These are exactly the kinds of strategies that become conceivable with this level of real-time monitoring. It's complex, no doubt, but it shifts the focus from just killing the bulk of the tumor to managing its evolution.
So the final thought for you listening is this. If we can now watch the evolutionary battle within a patient's cancer in real time, tracking which clones are winning and why, how fundamentally could this change treatment? What new strategies, what adaptive trials, what combinations might emerge when we move from reacting to resistance to actively trying to steer the cancer's evolution? That's the provocative future that tools like CloneSeq-SV seem to promise.